I'd like to start by thanking everyone, including departments, who answered my frantic emails to make this event possible. I couldn't be more excited. I have the esteemed honor today of introducing a truly remarkable person. A person remarkable enough to respond to an out-of-the-blue email from an eager, albeit over-caffeinated graduate student. Dr. Daniel Pauly received his master's and PhD from Kiel University in Germany and currently holds the position of Killam Professor at the University of British Columbia. Following his graduate studies, Dr. Pauly spent 15 years working in Manila at the International Center for Living and Aquatic Resources Management, where he worked extensively on development of methods that characterize fisheries statistics, such as growth and maturity, based on length-frequency data. He is the founder of the Sea Around Us project and has served on many editorial boards and NGO advisory boards, including but not limited to Pew, National Geographic, and Oceana. To some, perhaps what Daniel Pauly is best known for is the development of FishBase, the online encyclopedia of fish and fisheries information, including over 33,000 individual species, which receives over 600,000 monthly visits. To others, his work can be found in just about any ecology textbook. A prolific writer, Dr. Pauly is the author of several books and over 500 peer-reviewed papers. Google Scholar suggested the total was 916 articles, but to be fair, they included his 1984 paper titled, "Who Cites Your Publications When You Work in the Tropics?" Well, I would say quite a few, as he has over 76,000 academic citations, all of which cannot be Ray Hilborn. Dr. Pauly notably coined the concept of shifting baselines in 1995 and authored the highly acclaimed and debated paper Fishing Down Marine Food Webs in 1998. Called an iconoclast by the New York Times, Dr. Pauly has dared to step into the global picture of fisheries rather than the more traditional provincial view. To this end, his readers have been entertained with some of the highest stakes and most entertaining academic breakthroughs and debates in recent memory. Are we fishing down marine food webs? Does harvesting forage fish affect their predators? And will we see smaller fish as the ocean continues to heat? The latter of these, I presume Dr. Pauly will discuss today. To list all of the awards Dr. Pauly has received would be exhausting, but to put it all in perspective, he has 7 honorary doctorates. With that, it is my great honor to introduce Dr. Daniel Pauly.
You cannot imagine how glad I'm to be here, because it was a question of 1 minute, the connected flight between the red eye from Los Angeles and in Philadelphia. I had to run. I'm 71. I don't like that. I managed, and I'm glad to be here really because of you, because you're nice people, and also because this is the first time I really I get back to this theory, which you will see goes back to my dissertation a long time ago. I will introduce it two ways to you. The first way is this paper. Indeed, in 2012 or 2013, we published a paper based on previous work that we had that was global in nature about fish becoming smaller because of global warming. And basically, it was straightforward. All it did actually is, given that the gill of fish grow with a power of length that is around 2.4, and the body grows with a power of length that is about 3. The bigger the fish get, the less oxygen it has per unit weight. It is well known. It's almost trivial. But what it means is that as the fish get bigger, all of them, there is no exception here, the gill area per body weight, and therefore the consumption of oxygen per body weight declines. There must be a point at which what comes in and what is needed balance, and you can define this as maintenance metabolism, and and that's how big the fish can get. Now, if you increase temperature, fish are cold-blooded, we say that. Actually, they take the temperature of their environment. The oxygen requirement increase. We'll get back to that later. And therefore, the fish have to stay smaller. Nobody will want to argue with that, right? It's quite okay. It's trivial, and it should be. And this is a model that you can easily learn about and use it and play with it in your head, at least. Now, what about the evidence? This is the trout. This is a Japanese work. This is fantastic. This is a carp, sorry. And here you see the slope. It's 0.8. So 2.4 the length, the power, and 3. And the result of it is a slope of 0.8 that links Gill's oxygen supply and the body, 0.8. Obviously, the fish, when they are larval, the larvae, they have a little head, and they grow inside their little head faster. So larvae go faster, and the exponent is actually bigger than 1. But it's only for very small fish. And you know that from the lab studies that larvae grow exponentially, which means they are not limited by oxygen. They are only limited by food. And if there is enough food, they grow exponentially. If there is not enough food, they die. But after that, after they get into 0.8, after the limitation sets in, they don't grow exponentially, they grow asymptotically. That we all know fish grow asymptotically. They go toward the maximum size. Now, and you can see the pattern that I showed the previous image in here. The gill area becomes small, per body weight becomes smaller and smaller, and if oxygen, if that is the amount they need to attain this size, then when you heat up, they will have this size. No problem there either. There is evidence. And then what? This paper that we did was kind of routine, and all of a sudden, a group of 3 physiologists said it's all wrong. And basically, the evidence that they had is here. Basically, they say gill cannot be limited because the fish can generate as many lamellae as needed, and there is no difference between the oxygen supply and oxygen demand because of this. The fish can always produce more lamellae. And our conception, they say, is a conception that the gill match a sphere. There is no sphere in any of our paper, but you understand that the surface area of a sphere increase with a lower power than the volume of a sphere. So fish don't have this problem, they say, because you can always produce more lamellae. And for illustration of this, they produce that in the paper that that is a small fish. It has small gill, and that's a bigger fish, and it has bigger gills, and therefore there is no problem. And they can maintain the same gill area per body weight ratio. Now, I'm not going to lie, they really did that. And this is redrawn. That's the image you've just seen. And if we assume that this is a schematic representation of the gill area, this fish happens to be double as big as this one. Now, if it is twice the length, it's 8 times the weight, because it's a volume, and the third power of 2 is 8. So if they have to maintain the same gill area ratio per body weight, they need 8 times the body weight and not 4 times. This is a graphical error. You're messing up, you're presenting evidence that is false. I don't know if they did it by mistake or probably by mistake, because they don't have an intuition of what it implies to maintain the same surface volume ratio. And so let's be serious. The gill, that looks serious. This is a car that looks like, this is an old traction avant. This is an old Citroën. You see that in a French movie from the 50s. And you will note that it has a big radiator. And in fact, if you imagine a sports car, they all have big radiator sport cars. This is how you suggest, in fact, speed and power. Now, a sports car has an engine that needs to be cooled. And the hot water comes in from the engine, and it goes to lamellae, like gills, and it is cooled by air, and the air is heated by the hot water, and it's warm when it comes out, and then after all the water have gone through this lamellae, the colder water goes back to the engine. Now, it wouldn't make any point putting another radiator behind the radiator that you have. So this radiator, if you want to make it bigger, can grow in width, in height, but not in depth. There is no third dimension in exchange system. Now, this is explained here. You cannot put a third dimension in the exchange system. And in fact, you cannot, because there is no oxygen. If the exchange system is gills, there is no oxygen when the water comes out of the first layer of lamella. So in fact, it's the same system. You have hot water, cold water getting in. You have hot air, cool air, hot air. Here you have water and oxygen getting in, and it is then oxygen poor. That is, that there is only 2 dimensions. There is no a third dimension. So gills cannot grow as fast as the volume. And you would not believe, but so we have written to this paper that criticizes a refutation. Now they have again written a letter to the editor, and we have re-explained it using as an example that they don't get the dimensional argument. Now, that was the first way to introduce Gill limitation theory. Another way is to go historically about it, and it starts with me with a pink hat, and I just had gotten a master, and I was sent to Indonesia after I learned Swahili. Those of you who learned, I was taught Swahili because I was supposed to work in East Africa, and then I was sent to Indonesia. So we were supposed to assess the stock of the Java Sea or the South China Sea in 75. And that's one of the papers that came out of that. And we sampled of this station. This is Singapore, this is Borneo. And we had catches of that species that were very low. This Nematode species, doesn't matter what it is. This was the most abundant species in the most abundant family, and it was 1% of the catch. what do you conclude there? That actually you cannot assess stocks as you do in the Gulf of Maine, where there is what, one species or two, right? There were hundreds of species coming out on deck, and they were in part not even described at the time. And we had no identification books and material, no fish base. And the most abundant one were not abundant. So I decided to understand growth such that we could generate growth curve without studying the otoliths, and there was another reason where this was needed, because fish in the tropics don't have annual rings. If the temperature difference between summer and winter is below 2-3 degrees centigrade, they don't form annual rings. They have daily rings, of which I will speak later. So I I did my fieldwork in the library of the Institute of Marine Science 1 year, only reading and recovering all growth curves I could find and estimating the parameters of this growth curve. And I published a report that is actually well cited, where this 600 species that had growth parameters were available. That was the database. This was a database. And, and it was at the origin of fish base. Those of you who study fish will know fish base. I don't have to elaborate. But the first thing that was clear is that we have regularity. The von Bertalanffy equation, which described the growth of this fish, have 3 parameters: asymptotic size, asymptotic length in that case, a parameter called K, the Brody coefficient, and a parameter T0 that orients the length with vis-a-vis the absolute H, vis-a-vis the origin of the curve. We don't need the origin of the curve. The 2 parameters K and asymptotic weight in that case, length or weight doesn't matter here. You take log on both sides and you plot the growth parameters on this. And what you will see is that fish of a certain species occur in ellipsoid. The different stock of a species occur in ellipsoids, and the position of on the curve actually tells you whether they're fast or slow growing. That's what I did, the drawings I did with China ink, and that's what we do now with fish base. We just dump all the growth parameters we have. That is, for example, tilapia in aquaculture. They grow faster than fish do. This is a whale shark. This is the biggest. This is guppies. Here is rockfish. Here is cod. Here are, for example, the groupers. Tuna. Here are jellyfish, tuna, and they occupy certain part of this plot. I call them oximetric. Oxin is growing in Greek, and metrox is measure. So it's gross measuring plot. And the position of fish in there are clearly not determined by how much food you give them. They are constraints, anatomical constraints, that make fish be here rather than there. For example, if you take groupers, they are here. They are slow-growing big fish, and the same redfish, the Scorpenidae, they will be here, and here are the tuna. And this is not the question of the food that they have. So what is it a question of? And if you plot a gill area you will have large gills here and small gills here. By the way, these pointers are useful to detect whether people drink or not, because they tremble, the people who drink. So the first things that seem to affect where fish are within the ellipsoid, that is temperature, latitude here. Here is from a massive book about squids, or the squids that occur in the north area are bigger for the same species than the squid occurring in low latitude. This is a phenomenon that we'll find again and again in fish, in fish and invertebrates, that they are bigger at where the temperature is colder. So now I must bore you with a model of how fish grow. And if any physiologists, you may not talk, only at the end, if somebody knows better. So we have the growth of the animals, the growth rate of the animals, that is the slope of the growth curve. And this growth will be the result of two processes, one positive, the other negative. The synthesis of protein and other body substances is an oxygen requiring a process which, because it is oxygen related, is not proportional to weight, but to weight to a power that is related to the gill area of the fish, because the gill area limits the amount of oxygen that comes in the body. So this process is not the synthesis of body substance, is connected with weight, but with weight to a certain power. And yes, I've called it D because of Daniel. So the next, and this power, you can see different fish have different powers. And von Bertalanffy, the person who described the von Bertalanffy equation, who made this equation, he did experiment with guppies. And guppies happen to have an exact isometric growth. The gills or the respiration scales with a square of the length, and the body weight with a cube. And so the ratio in guppies is exactly 2/3. And von Bertalanffy made experiment, proper experiment, extrapolated an equation, got an equation with 2/3, and we use this equation because Beverton and Holt, the fathers of population dynamics, took this equation. However, because they knew that in other fish, in fish other than guppies, you measure the respiration, or the gill area is, well, the respiration is not, doesn't scale with 2/3, but 0.8, 0.7. They describe the von Bertalanffy equation as an empirical equation, not a physiological equation. And you can look at various fish. In general, fish that can become big, like tuna, have gills that scale with weight very close to 1, 0.9, tuna in particular. And fish that become very small, that stay very small, these tiny gobies that are 1 centimeter when they're adults, they have very low values. In fact, this value, this square, was produced later, came later after this curve was published the first time. So that's the range of values of the exponent that exist in fish, with some exception. One exception is larvae. Again, when larvae are tiny, they gill grow faster. Their respiratory area, actually, it's not only the gill, because they respire via the skin and via the primordial fin fold. and they have gills that grow with the power of 1 and more. So for this reason, they grow exponentially. But other fish don't grow exponentially, and that's because the value of the scaling factor of gills to weight is less than 1. So on the other hand, the catabolic process, the negative process, are weight dependent. The catabolic process are much misunderstood. People think this is the complete utilization, the burning of substances, which would require oxygen. But actually, a molecule that is in a body, in a living body, has a primary structure, like the sequence that DNA has. That's a primary structure. Then they fold, and this folded structure folds again and folds again. They have a quaternary structure that makes it possible for any substance, protein and stuff, to work as an enzyme. It locks in a receptor because of its shape. Now, every molecule has living molecule has a half-life that they don't last. The quaternary structure is very fragile, and a small shock with another molecule breaks it. And once it has lost this quaternary structure, it actually has to be resynthesized because it doesn't work anymore as an enzyme. So that is called denaturation. And denaturation doesn't require any oxygen. It occurs in all cells of the body, and is strongly temperature dependent. Because what is temperature? It's actually the intensity of collision, right, with molecules hitting each other. So this process is temperature dependent, but not oxygen dependent. So you have this growing with a power smaller than 1, and this growing with a process equal to 1. So once the amino acid have lost their structure, they become part of the pool of amino acid in the body, which also includes food. And these amino acids are used as fuel for energy metabolism, which fuels protein and various activities. Now, if these activities take over, and if temperature increases and increases activities, there is less for protein synthesis. So basically, gill size, on the other hand, gill size doesn't increase. It increases with size, but it doesn't increase with activities. So you have a problem that it's bigger the bigger the fish get. And that is independent of the O2 content of water. This would happen anyway, even if the oxygen content of the water remains the same. But actually it decreases when temperature increases. So you have a double whammy hitting the fish that grows in warmer water. And that gill size modulate energy metabolism is obvious because Fick's law says that the water distance is that oxygen that goes through gills is strictly dependent on the surface area of the respiratory area. It's also dependent on the thickness of the lamellae, the so-called water blow distance, and the difference in inside and outside the gill lamellae. But this is additional factors that don't necessarily change, but gill size change with the power of D. And this is lower than the power of catabolism. So the fish have to grow asymptotically. The growth rate gets smaller and smaller and whatever the ration is, whatever the food they get. So that's the story. Yeah. And it's true. If this thing is true, if this is true, and my interpretation of it is correct, this is the reason for lots of things. For lots of things, and for all animals that breathe water. It's not a fancy that I have for this fish or that fish, or for something in the Gulf of Maine, but not in Indonesia. It applies everywhere. I formulated that in my dissertation, and it was so foreign to the thinking of my colleague at the time. I didn't dare to work on it all the time. So I wrote papers and papers on the side about that, and my work is about stock assessment and fishing downwind food webs and fish base and ecopath and stuff. But actually my real passion is this, because this is a real scientific theory. The rest is what's and that's so. What do you call this? Circus. More. That is real science. Now, the reason why this is science is because it explains more than I started to explain at the beginning. And an artist drew for me the watch that you're going to see, and we went back and forth with these watches until I got exactly what I needed. So the basic equation is here, and obviously the first thing is that asymptotic growth is evidence of that theory, but it's the evidence I've used to derive it, so it doesn't count. All right? Now, no, this is not by accident that this stuff is written in Gothic letters and is inverted, because you're not supposed to be able to read it. But I will tell you what it is. Why do female fish get bigger than... Okay, let's turn it around. People say fish stop growing when they spawn. This is marvelous, an explanation. It sounds good, doesn't it? But then, about females, 70% of all fish, the female grow bigger than the males. And now, their energy expenditure for spawning is much bigger than the male. In fact, this is how you define a female. If you don't know, if you went into space and there would be some animals on Europa or Enceladus in one of these ocean moons, how would we differentiate the female from the male? By definition, the female have more reproductive investment. And yet, the females are bigger. And if you saw this in terms of weight, it would be much bigger. The idea that fish have lots of growth when they don't reproduce, and when they reproduce, the growth is halted or diminished, is wrong. Because if this were true, the female would always be smaller. And in fish, they usually are bigger. So the solution is actually quite simple. The females are a bit quieter. They are not. So they are more quiet. The males are fighting for access to the females, and they are producing colors and behaviors and stuff. That's why they say smaller, because they use the oxygen for something else than growing. And in fact, in aquaculture, the first thing we do is modifying the behavior so that they are quiet. When they are quiet, they grow better. Compare a salmon that is raised in a farm with a wild salmon. The big difference is actually behavior. When we breed fast-growing fish, we breed calm behavior. That's all we do. And that's the reason why females are growing bigger than male. Now, second, the food conversion efficiency of fish declines with size. That's known for everybody who works on this. In fact, you can invent an equation. They usually use that equation to describe the food conversion efficiency. In other words, the growth increment divided by the food eaten. But this equation is better because it includes a maximum weight. And if you do that, the food conversion efficiency, you get a fit that is similar to the fit of the standard equation, and you get W infinity. So you can actually show that W infinity is simply the size at which a conversion efficiency gets zero. That is, they eat food, but they don't grow anymore. Why? Because this food is just what you can process with the oxygen you get. So you can give them food in excess, but they don't convert it into growth. So that is, mind you, is a mystery. There is no good explanation why the food conversion efficiency in fish decline. Now, that is what. You see the little watch here? Please like them, because lots of people, it cost me quite a bit of money to have this watch moving with every hour. Now, this is wild, but think, why do fish spawn at a given fraction of the maximum size they will reach? They cannot predict the maximum size they will reach. And how do they adjust to the fact that in certain environment they're going to be smaller? How do they know that they have to spawn before? I say it's because the process that makes them smaller is also a process that makes them spawn. And conceptually, if you remember this concept there, if they reach W infinity here, they have to spawn at a certain size here. This is smaller. And this then is the metabolism at spawning, and this is the metabolism at maximum size. All right? Now, if you increase the metabolism because you put them in hot water, they have to spawn at smaller size, and they do. And the ratio between these two things is the same in all fish. That is tuna, and this is guppy. The ratio is 1.4, and it applies to all fish. Basically, all fish have, that is down the hypothesis, have a sensor that tells them, I'm getting too often out of breath, I have to spawn. Mind you, this morning I was running in the airport of Philadelphia, and I didn't think about spawning at all. But if you begin thinking about it, there is lots of instances when stress induces spawning. If you increase temperature on a bivalve, they spawn. That's this process. I have not made such a plot. The paper that published that, all of this is published in the primary literature, was reviewed by 5 or 6 people who all hated it. And the editor sent it again and again because he wanted to know what is it that makes it impossible for people to understand. And in fact, it's very easy to understand, right? When you get bigger as a fish or any air breather or water breather, you get into oxygen trouble. So if the trouble are measured, the same way, for example, that carbon dioxide is measured by a sensor that is in our aorta, the same way you have to imagine if they integrate over time, lack of oxygen, I'm getting in trouble, it's time to spawn. If you imagine a thing like that, it would be conserved because all fish need it, and it would be the same because all fish have it since whatever the Devonian finfish exist. I would like somebody to pick that up, because there is one substance which is used by all fish as an indicator. But no physiologist has ever bothered to look at that, even though it explains why fish spawn when they do. Next, that is a bit more elaborate. There is a process in fish called abortive maturation. They spawn, they mature, but then they don't spawn. The eggs are resorbed and they wait for the next season. Now, that is a mathematical concept called the cost catastrophe. It comes from topology. And it says that a factor 2 and a factor 1 can combine to generate processes which at certain level do not change qualitatively, but at certain other level change qualitatively. And if you have the year here and the size of the fish, you can have a fish that goes through the season without noticing anything. I remember my son at age 8, he could see a supermodel walking in front of him and he would not notice anything. At 8. At 9? Nah. 10? Nah. But at 11, 12, he would begin to notice. Something has happened inside of him that makes him notice something that happened outside regularly. So, at some size, you notice there is spawning season, but you don't get over the hump. And then spawning, you are over the hump, and you might actually get into a hysteresis loop. So that is spawning as a cost catastrophe. It explains something that other colleagues explain as a group phenomenon. They say that it is evolutionary sustainable, evolutionary stable strategy. But it's not sufficient to show that something works for the population as a whole. You have to explain it for individuals, because the population as a whole is the result of individual motivation, individual acts. And you still need to explain what motivates a single fish to spawn. Anyway, so that, and then there is this thing that fish are bigger at the closer at the cold end of the temperatures, and that is clear. This is you have a lower O2 demand, and which allows you to grow. This is a phenomenon which physiologists don't care about, but actually it is observed hundreds of times. There is a huge literature about it, and a theory of growth must incorporate an answer to that one. Well, also we know that the big old fish of a given population are always in deeper water and colder water. That's the reason. And the evidence, when I was assembling this, I left one of the watches. It was supposed to be here. So that is a mess up. If you ever use this thing, please remove it. So this is Heineke's law. This is a German scientist who said if you go into deeper, colder water, you get a larger place, a few of them. And this is the observation simply. Two fish from Hawaii, they are bigger in colder water. Now, growth and food conversion efficiency, I mentioned, are reduced when the the water content of water declines. The aquaculturists, the people who grow fish in ponds, in catfish in Arkansas or in China, there is lots and ponds, thousands, millions of ponds where people grow fish. They aerate the ponds. And there is a whole technology on how to aerate a pond and how much energy is needed to force air into the pond. And there is no theoretical connection to academics, physiologists who could tell them, well, you're doing that because you don't want the fish to run out of oxygen. So there is no, the practitioner know that they have to put oxygen into their pond, not to prevent death, but actually for the fish to convert the food they get into growth. Because otherwise they can't. If this is too hot or there is no oxygen, they will eat, but then they excrete the stuff out of their gills, the amino acids. Fish often perform large-scale migration, and the amplitude is higher in a big fish. And it's clear that salinity temperature change force the fish to migrate. This is West Africa, and this is Senegal and Gambia. This is Morocco, and the fish. This fish makes migration. I have a better version of this. This is the month. This little scribbling here. If you translate this into temperature, the fish, and you have latitude here. The fish actually, all they do is stay in the same temperature. That's why they migrate. So, temperature is actually explaining migration, not food, not other stuff. It's simply temperature. Finally, no, we are 1:45 here. The center of distribution of fish are moving poleward. You know that, and we have lots of papers on that. And in temperate areas, you have temperate fish and a few kind of subtropical fish, and the temperate fish are disappearing. They are replaced by subtropical fish, and in the future there will be only the subtropical fish. That's what happened in Maine. It's on the way. It's happening. In the tropics, when you start with tropical fish, you will have fewer tropical fish and fewer tropical fish. That because there is no hypertropical fish to replace. This is also explained very nicely by extreme sensitivity to temperature that is not replaced by food or something. The fish don't have the option of choosing between lots of food and bad temperature or good temperature. They have to follow the temperature. And if they find food, that's good for them. But if they don't find food, they cannot not go where the temperature is right. We can. They cannot. Next, the pH in the muscle of fish increase in fish that are fighting because they cannot get enough oxygen into the body when they are being played. The pH can drop enough for an irreversible reaction to be initiated. Essentially, the fish goes into autolysis, and that is well known. This autolysis, it leads to so-called burnt tuna, and also fish that have fought long time in hunger. They are not good to eat. That's the reason, because essentially the vacuole that contains enzymes, digestive enzyme in cells break, and the fish become a low pH thing. This is also what happens with us when we run in airports, right? Now, another one that totally unexpected, it's almost finished. Daily wings. Daily wings, you have heard of them. Have you heard an explanation ever why they are formed and how they are formed? Never. Never. You cannot find outside of an oxygen limitation theory an explanation for them. This is, if you start with this thing here, in reality, the line is not straight. The line varies as a function of activity level. If you're very active, you use up some of the growth, some of your growth potential which is there. And if you are very inactive, it's available. So in a course of 5 or 6 days, this happens. And when you're very active, there is no free oxygen in your body, and the pH drops. Right? And you're very inactive, you're quiet, the body is flooded with new oxygen, lactic acid is eliminated, and you have this. Now, daily wings, the people who study them, they occur in all kinds of animals. They occur in corals, but in fish, you can see them only in larvae and juveniles. Why? Because here, the fish are always in oxygen trouble. They don't have much scope for oxygen, and at that time, the otolith look like blank because they are always in the same regime, which is out of breath all the time. Whereas the larvae really recuperate at night, and they at night there is an organic rich layer produced, and during the daytime it's etched. It's etched by the low pH. Now, how do I know that? Because some people have followed the variation of pH inside the little capsule within which the otolith is, and they notice this fluctuation. They have also done that for squids, and I didn't want to put in each slide the papers, but I will at some point do it also. And I have developed this theory over times, and people cannot intuit. They cannot. It's not intuitive to us because we are mammals, and breathing is for us very easy. Now, if I do that sometime with students. I give a straw to each student, and I ask them to breathe through the straw, and that's being a fish. You have to breathe through a straw, and when you breathe through a straw, you can see how difficult it is to have a low oxygen supply. You have to be very careful with the oxygen. You have to be not excited and stuff. You can run a few seconds or minutes without it in anaerobiosis, but you then need to recuperate. And I have only a few colleagues who have understood this theory, the subtle aspect of this, and one of them is Andy Bakum. He's an oceanographer who thinks dimension, who can think like a fish. And he's the only one who has taken on to develop that further, and who has, in this late hour, which start again, been able to bring it further. And if you look at a beautiful movie about the ocean and stuff, you will see very often a predator and other fish living in what appears to be harmonious relationship. The predator doesn't eat, doesn't lunge, and because if they do that, within seconds, they run out of oxygen. And if they run out of oxygen, they cannot move. So the super predator can get them. So this one could eat that fish. but it would require the chase that would be required for that would make this fish vulnerable to the shark that you cannot see but is there. So he, Andy Bakhun, says that the oxygen constraint is actually a reason why you see this seeming harmony. So you have 12, I have given you 12 good reasons, of which 11 were not used in the derivation of this theory. Now, there is lots of things but they don't fit on a watch, on a clock. That we could talk about. For example, what happens with the air breathers? Well, have you seen an arapaima from the big fish in Amazon? A big fish occurring in the tropics? That is a contradiction. No, this is an air breather. So the arapaima, if you prevent it from breathing air, will drown, will die, because a fish that big cannot exist in the cold water of the tropic, in the warm water of the tropics. Now, how about whale shark? Very hot water, and it's the biggest fish in the world. Actually, whale shark are always diving into deep water to cool themselves. Okay, some colleagues have worked on that. Now, what do we demand of a scientific theory? It should be based on a vast body of observation. Its central tenet should have been independently tested, which was the case in the case of this thing. It should apply to aspects of the biology that were not used to derive the central hypothesis, and it solves a number of puzzles that were previously explained with just those stories, because each of these 12 things and more is explained by individual explanation, ad hoc explanation. It's like if you look at global warming, the people who object to global warming, the 2% of scientists or whatever, they themselves have different explanation. So they have just those stories. One of them says because the sun, and another one says because the Chinese oaks, and another one. They all have different stories. Whereas the consensus of most scientists have one story. So it is important is that you have a broad hypothesis, a broad theory, as opposed to just a bunch of ad hoc, unconnected ad hoc stories. And finally, there is the U.S. Academy of Sciences says that a scientific theory is a well-substantiated explanation of suspect natural world that can incorporate fact, laws, and inference and test the hypothesis. And I'm claiming that oxygen limitation theory is a theory, and it is actually the best available nowadays to explain things that many people think are not even questions, that they don't see the question. But actually, there you have, they are questions, and you have the answer. Thank you very much.
Do we have any questions?
I'm curious, when you were talking about the trends, the data you're showing out from a number of species and such, and things like shape and size, as well as lamellar distance and metabolism, are all very variable across individuals within a population or species. And so would you expect to see the same sort of relationship among individuals in a species with regard to the relationship between
Yes.
the
Yes.
lamellar?
If you look at a stock of cod, say, we produce one growth curve for stock assessment. But actually, if you work on the individual cod, they all have different growth curve. And that's the reason why, for example, after 100 years of fishing, the average growth curve changed because the bigger one are fished first, right? So they have individual growth curve, which are reflection of individual temperament. If a fish is aggressive and is always fighting against other fish of the same batch, that fish will pay a cost in size, but actually it may pay off in spite of it in terms of reproductive output. So since people begin to look at individual fish, they notice difference, and the difference is what we work with when we breed fish that are fast growing, and that's also what we modify when we fish and select for the biggest one. We have after a few decades a bunch of runs. The lagoon in which I worked for my master, that was before Indonesia, had fish this size and tilapia this size that were being fished, and now they have become like guppies this size, and they produce at this size because there has been a selection against the big one always. So they're all different, all of them, but they're all subject to the same kind of law, the same constraint. Yeah.
Mark Wells, I think you're up first.
Yeah, very interesting. I work with plankton, so I'm interested in what size limit or threshold does your Gill-Oxygen Limitation Theory no longer apply?
Actually, in Calanus and other zooplankton, this fully applies. You can see that when the cold form of Calanus are bigger than the warm form, and they are seasonally also the size vary with temperature. And the point is that when you get to a small size, very small size, they don't need any organ to bring the stuff in. It's just diffuse, and then the diffusion rate is different for different nutrients. So it can at some point become another factor than oxygen. For example, I don't know, nitrogen diffusion into the body over phytoplankton. But essentially, the transition from big datum to small flagellate that is happening worldwide as a temperature increase is really another is related to this stuff, because this theory is really a theory about surface and volume tension. I speak in my book about that. I speak of dimensional tension. And dimensional tension, we have all kinds of them. For example, on land, where the problem is not getting oxygen into your body. The problem is gravity. And on land, the dimensional tension makes itself noted by the cross-section of the legs. If you take daddy long legs, a spider with very thin leg, you increase the size of the body of a spider, what do you get? A big daddy long leg? No, you get a tarantula. A tarantula has legs like an elephant. Why? Why do they have so big leg? Because the cross-section of the leg is a surface, and the volume is a volume. And the volume goes too fast for these filiform legs to continue. So this is gravity, and it's also heat production. If you take whales, the volume, gravity is not a problem for whales, but the heat production is the problem. They are hulled in fat that protect them against the cold. But then if they run between two terminals at the airport, they will get hot. And how do they get rid of heat? to their fluke, and because if they open their mouth to cool themselves, they slow down. So if they want to escape, they can get rid of heat only to their fluke. It's a surface. So the whalers, the Boston whalers, they knew about that, and they chased the whale, and they chased them to death. And there's the same thing in the San people in Kalahari. They run antelopes to death because they cannot ventilate. They cannot get rid of heat only through the tongue, and as opposed to people who get rid of heat over the whole body. And so we can run to death any mammals, except wolves and a few other. Even horses, we can run them to death. And this is a surface volume thing. But every medium has another dimensional problem. And our dimensional problem is that we get rid of heat. And therefore, we maintain a high temperature. We must eat all the time. And birds, it's even more so. And because of that, oxygen is no problem for us. Breathing is no problem. Our problem is getting enough food in our system. It's not mine, but it's... And we have then these glasses that we impose that on every other animal. But the problem of a grouper, of a large grouper that sits in a reef, is not getting food, is getting enough oxygen. If they start swimming, as soon as they have done a few strokes, they are out of oxygen. And that is the problem. And that's why, for example, a grouper feeds by just opening its mouth. And that's it. And if you want to chase your prey, as tuna do, you become mammals almost, with gigantic gills. What is the price tuna pay for the huge gill they have? They cannot live in close inshore water because the gill would get clogged up because the space between the lamellae is so tiny that they cannot afford to live in close inshore. So there is a price to pay for the gills that you have. And these gills that you have, people always ask me, but then why don't they have bigger gills? Well, they have bigger gills because if you look at Devonian fish, the gill apparatus was much smaller at antique fish, but fossil fish. But they grew enormous gill. You have the gill that you need to reach past the size at which you produce. But then they become a handicap because as you get bigger, they don't go as fast as they should. And so they become a problem for you individually.
All right. Maybe one really quick one. I know some people have to run different places. And really quick, also, we're hosting lunch at the Union after this. Anyone's welcome to come join us. Yeah.
Another cost that tuna pay, of course, is circulatory adjustments.
Yeah.
And I'm sure with the database you've got, you looked at it, there's a huge amount of numbers, and it doesn't change the basic tenet that you're saying. But I'm curious, how much variation is there just in terms of differences between gill surface area as a measure of oxygen supply and perfusion of that?
You see that as soon as it gets away from it being limited, because blood circulation occurs in a volume, all the things that are dimensionally scalable to weight, I don't look at them because they cannot be a limiting factor. Surface area is a limiting factor because it has to function as a surface. I don't know of any system inside the body that has a similar limitation. I'm not saying, and there is this thing called synmorphology. The different organ systems have to be adjusted to each other, but some of them can be easily adjusted, and some of them are adjusted at great cost. And the surface is always going to be a problem because the gills have to remain a surface. They cannot be stacked up in a third dimension. Whereas all the other organs that we have, from the prostate that is a problem for old people, to for old men, to whatever, these organs have 3 dimensions, no problem.
I agree, and I'm just curious though, within that, there ought to be some variance in, for a given gill size, can you predict what sort of fishes, if not
I thought you would notice that I don't know your answer.
what sort of fishes might be predicted to do better as it warms up for a given?
Basically the sharks and some big sharks and some have been able, because they have a high metabolic rate, to turn heat into an asset. But it is an asset if you metabolize faster. It's an asset that has a price, and that's the same price that mammals pay. We need more food. So it's the fish don't. And if I've seen Jurassic Park lots of biology in there. And the point about the dinosaurs is that they had a high performance like birds, but the rest of the time they can be quiet like crocodile. And there is no animal that can do that. Large predators that do that. The lions have to maintain their metabolic rate and the temperature at the same temperature, their body at the same temperature. So that use lots of energy, which a crocodile doesn't need. But then they don't have this pulse of energy that maybe dinosaurs or velociraptor could develop.
Let's give Daniel Pauly a hand, guys. Thank you so much.